Our next talk is the first of our tech talks, which these were sort of submitted competitively as abstracts, and then we chose a few talks. Is it not? It's not doing anything. Oh, I just need to be louder. Okay. Oh my God. Um, Anyway, these tech talks were chosen from competitive abstracts, and these are sort of our idea with this is to look for emerging technology or applications, novel applications, and so this was one of the cooler ones that we thought would be really valuable to show. Let me get that slide up real quick. They're just all called FlowTex, so it's hard for me to know which is which. Okay. All right, so our speaker today is Justin Dela Cruz. He's a senior scientist in scientific affairs at Solaris, where he leverages his expertise in cell and gene therapy analytics to advance automated QC release testing for cell therapy manufacturing. He focuses on connecting scientific innovation with commercial strategy to deliver scalable solutions for cell therapy developers. Before joining Solaris in 2024, Justin worked as a scientific solutions consultant at Benchling, supporting scientists in digitizing data management. His analytics career includes roles at Sangamo Therapeutics, Adverum Biotechnologies, Biomarin, and Ultragenics, specializing in analytical method development and QC testing for AAV-mediated gene therapy programs. Justin earned his PhD in microbiology, immunology, and molecular genetics from UCLA and completed postdoctoral training in infectious disease at Stanford University. And outside of work, he enjoys grilling and being a girl dad. So welcome, Justin.
All right, thank you for the great introduction. So I know I'm the person standing between you and a great networking session, so I'll try to get through my slides. Again, I'm Justin Dela Cruz. I am a senior scientist for Solaris, and Solaris, we provide services to automate and standardize cell therapy manufacturing and QC release testing. Today, what I'll focus on is about how we at Solaris are thinking about automating and standardizing QC flow-based testing as we expand globally. So a little bit of a switching gears here because we're thinking, how do we scale commercially beyond the clinic? So this presentation will highlight some of the key limitations around the use of biological controls and how we plan to address these challenges to enable fully automated end-to-end QC testing at a commercial scale. Let me grab my laser pointer. Okay. All right. So cell and gene therapies, as we all know, are curative therapies, and most processes today rely on donor-derived PBMCs or leukopak material for controls and assay startup. And those of you who thought about scaling up and manufacturing and who've worked with traditional CDMOs understand that, right now, currently, you could say that they're mainly manual, low-throughput QC testing, and that they, typically also lack standardized site-independent controls. And as you see here, there are many manual touch points, all the way from scheduling to sample prep to execution, analysis, verification, and final reporting. Both testing and analysis are fragmented and prone to operator bias, and that's very hard to scale. So when trying to scale both manual QC testing using biological controls, you can expect the following limitations. So we've heard it many times already that every donor is different, which means differences in marker expression, immune subset distributions, activation states, marker densities, and so forth. And that variability shows up as site-to-site drifts, operator dependency, longer investigations and CAPAs, and then slower tech transfers when you try to move methods between facilities. So as teams scale from one lab to, say, multiple global sites, that becomes a real operational and regulatory burden. So the result you'll invariably see is delays, variability, high costs, high failure rates. So at Solaris, our solution was to remove all of the manual operations by automating QC testing processes from sample receipt all the way through to final report generation while still using our clients' existing reagents. So compared to manual QC workflows, each step is designed for scalability and complete digitalization, all while being orchestrated by a system schedule to ensure maximum efficiency at commercial scale. So I'll start with the ending first and just say, what Solaris is able to achieve is that instead of QC being site-specific and very manual, it becomes much more standardized, scalable, and plug-and-play. So what that means is faster tech transfers between sites, analytical comparability between sites, reduced regulatory risk, lower operational burden, and real-time monitoring of materials and hardware. So this model here is foundational to both our manufacturing and testing as a way to build reliable global automated cell therapy manufacturing production. All right, so you might have noticed the large square in many of those slides. So I'll try to walk you through that right now. So to achieve true high throughput QC testing, it's not enough to automate just a couple steps here and there in the process. So a system like the Solaris CellQ, which is what you're looking at, is our platform, which brings together flexible architecture, intelligent software, and tailored engineering. So I don't take any credit for this. I work with a lot of brilliant automation engineers that built this from the ground up. So let's take a quick tour of it. When QC samples are handed off to the CellQ platform, that platform coordinates the reagents and assay materials, automatically loads them, initiates the automated workflows, and even launches system and process verification checks to automatically confirm that everything is ready. So we have unique 2D barcodes on samples, on consumables, on all the supporting reagents to maintain that chain of identity and chain of custody throughout. The supporting workflows like reagent and waste management is also fully automated and managed with lot control and expiry tracking. Each assay has dedicated pre-kitted solutions, so we've actually collaborated with a number of suppliers to pre-kit their reagents into our automation compatible containers, which I'll show later on helps reduce time even more to set this up. Final assay plates are prepared by a automated liquid handler and then transferred to the analytical instruments by a robotic arm. And then our instruments, so we use off-the-shelf instruments that are best in class, so we didn't reinvent the wheel by making the instruments as well. But these instruments are seated on what we call a modular cart system, which is designed to maximize uptime. So if you imagine any analytical instrument goes offline for maintenance or what have you, another pre-qualified cart with the same instrument seated on it, complete with its own computer, backup power connections, can be rolled in, swapped, powered, and re-networked in under an hour. And then finally, data is captured and directly transferred into our LIMS platform. So from there, results are reviewed, signed off, and a certificate of testing is generated. So our internal software controls this QC submission process, method order creation, recovery workflows, and full connectivity to our Solaris ecosystem. So I was very excited to hear, our panelists earlier today kind of name off these different wish lists of things that they would like to see implemented. And I'm happy that that same wish list is sort of recreated in our QC platform. Okay, so a key enabler of repeatable QC testing, and I emphasize repeatable, is the use of standardized controls. And so the question we're asking here is quite simple. It's where can additional standardization help reduce risk and make QC easier to scale? So in this study, we looked at standardized synthetic cell mimics as one example of that strategy, especially for more complex assays that are flow cytometry based. So again, biological controls, we're all familiar, tend to be flawed, can result in variable data quality, are complicated in terms of sourcing of donors and characterizing a material, and at times unavailable due to rare biomarkers or rare disease indications. So at Solaris, we're also, I should say, we're reagent agnostic. But that said, we're always evaluating innovative reagents and instruments to make sure that they're compatible with our technology. So that's why we evaluated reagents from a company called Slingshot Biosciences, who have developed these polymer-based cell mimics. They also, by the way, have a booth in the other hall, so go visit and talk with Natalie. So these cell mimics can be off the shelf. They can be customized to have defined scatter properties, stable subset ratios, controlled antigen density, and manufactured to specification, to name a few. So we ran an initial comparability study using Slingshot's TrueCytes lymphocyte cell mimics as our controls. We evaluated multiple lots with different operators, different days across different instruments, and across different labs, and directly compared performance against traditional PBMC controls. So we used their cell mimics as a sort of model system because they're well characterized and consistent, which makes them a good stress test for automation and site-to-site comparability. So real quickly, a search of the literature helps us to benchmark the variation we can typically expect from healthy donor PBMC. And so on the left here are the lymphocyte subsets that we focused on for these experiments. Cytotoxic and helper T cells, B cells, monocytes, and NK cells, or TBMNK for short, rolls right off the tongue. Immune phenotyping by TBMNK is a standard QC release test for any cell therapy product, which is why having a repeatable and available control for this method is important for automation, for scale, and for delivery of treatments to patients. So our in-house data confirms that in testing 3 different healthy donor PBMCs, we showed substantial deviations from these benchmark values and also high variability from donor to donor. Here we compare our measurements of each lymphocyte subset in the TrueCytes mimics to the percentages certified by the manufacturer. So using the cell mimics, the differences we observed over here on the right were generally single digit, with our highest percent differences being only 8.2%. So right away, by using cell mimics, we can remove one of the biggest sources of analytical noise. And visually, it's even more telling. Beyond the summary table from the previous slide, these representative flow plots make differences very tangible. On the top row here, you're looking at healthy donor PBMC, and you can see that the populations are more diffuse with less distinct boundaries between the subsets. And then on the bottom row are these cell mimics, where the populations are much tighter and much more clearly separated. So visually, you can appreciate that you get less scatter, cleaner clustering, which in theory makes gating simpler, far less operator dependent, and enables concepts like automated gating, automated analysis, a possibility. If you already do automated analysis today, I'd like to talk with you. So another source of analytical noise can be the instruments themselves. So here we demonstrated that instrument-to-instrument equivalence by using a calibration reagent called Scatterbridge, also developed by Slingshot Biosciences. These plots show an overlay analysis of calibration reagent data from two independent flow cytometers, and where they overlap in green. I wish I knew the beautiful color palette schemes that were presented yesterday. I asked Claude what to do here. So this is the color schemes it suggested. A simple performance verification check like this prior to an experiment, you can leverage that to eliminate flow cytometer variability as a confounding factor in multi-site comparative studies, and then also enable more direct cross-site comparisons of these true site performances. All right, now that we've standardized the controls and removed instrument-to-instrument variability, what can we expect out of intermediate precision and inter-laboratory reproducibility? When we compared data testing the two different lots of mimics across instruments, scientists on different days, different labs, the cell mimics again showed very tight clustering and low CVs. So practically speaking, the results look the same no matter what where the assays are run or who's running the assay. And that's exactly what you want for tech transfer and global deployment of these assays because you're no longer chasing whether the differences are due to the method or due to the starting biology. So the broader takeaway here is not just that maybe cell mimics are better controls, but it's also that when you introduce standardized, well-characterized reference materials, you can dramatically reduce variability and make complex assays much more portable and automation friendly. So today I talked about the benefits of standardizing QC testing through automation, but also the benefits of leveraging synthetic controls like cell mimics. So what happens when you combine the two? This is our big next steps. But what we think is it can significantly reduce the time it takes to source the controls. So instead of taking several months to screen for donors, characterize the controls, document them for use, we can simply automatically reconstitute off-the-shelf TB NK cell mimics in minutes. And we're actually working with Slingshot to lyophilize these mimics in our automation-friendly tubes so that we take away that extra step that's a manual step. And then again, testing of the samples. So instead of taking hours to manually verify that your instruments are ready, that you thaw samples uniformly and staging supporting reagents, we can initiate true walkaway QC testing, analysis, and report generation in minutes. All right, so finally, Solaris is planning facilities in multiple countries and continents, so it's imperative to ensure operations across these sites are identical and predictable. So standardized tools like cell mimics are optional, but we use them when they add value, such as when customers want higher comparability or more hands-off automated QC testing. We can use them to calibrate instruments, and they're especially helpful for complex assays like flow cytometry, where donor variability may otherwise dominate performance. So by combining these consistent materials with automation, we can enable faster tech transfer, lower training burden, operator dependency, and have things like more verifiable instrument readiness and more predictable performance across our sites. So that's my last slide. I'm happy to take any questions.
Okay, still on. Thank you for the nice talk, and definitely worthwhile after what we heard this morning about trying to standardize stuff. Just curious, where kind of like throughout it's cool that you're trying to automate basically the entire system. What are the kind of major pain points that where do you need basically human checks through that, or
In flow-based assays specifically?
yeah.
So that's a great question. So in the middle of the talk, I blurted out if you do automated analysis, that's talk. That's really one of our most manual interventions right now is still the analysis. So the analysis that I showed was prepared manually. So it's our aspiration still to automate fully end to end. But that analysis portion, we still need scientists for that data interpretation. Even if we do automate that at some point, it's still important for us to have the scientists there still to interpret and troubleshoot.
Thank you. Any questions? All right. Thank you, Justin.